Okay, hello biology, this is Mr. B, and we are continuing on with cell boundaries in Chapter 7, Section 3. So this is our last day with cell boundaries, and we talked about diffusion and osmosis, about how those were processes that did not require any energy and allowed things to move across the cell membrane. So now we're going to get into some things that do require energy. So we talked about regular diffusion. The first thing we're going to talk about is facilitated diffusion, and cell membranes have protein channels that act as carriers, making it easier for certain molecules to cross. So what that looks like is, what that looks like is right there. So that little pink mass here is a protein channel, and the glucose molecules is an example of something that uses this protein channel to pass through. And the movement of specific molecules across cell membranes through protein channels is known as facilitated diffusion, and hundreds of different protein channels have been found that allow particular substances to cross different membranes. So facilitated diffusion, if you think of like a quarterback, and a quarterback is like the facilitator of the football team, and the point guard is the facilitator of the basketball team, and that just means they provide help. They kind of get everyone else involved. They help everyone else out to do their best. So that's what facilitated diffusion is. It's diffusion that has some help. And then again, there's the example again. There's millions of different, or not millions, hundreds of different protein channels that are specific to different molecules. Glucose is just one example. And then although facilitated diffusion is fast and specific, it is still diffusion. Therefore, facilitated diffusion will only occur if there is a higher concentration of the particular molecules on one side of a cell membrane as compared to the other side. So even though we're using this protein channel right here, it doesn't mean that it can go from an area of low concentration to an area of high concentration. It's still only going to go high to low no matter what. Even though it has to go through a specific channel, it still only goes from high to low. So sometimes we're gonna go against what this says. So sometimes cells move in the opposite direction from which the materials would normally move. That is, they go against the concentration difference. So they go from area of low concentration to an area of high concentration, and this process is known as active transport, and active transport requires energy. So active transport is going against the concentration difference. It is going from an area of low concentration to an area of high concentration. And another name for active transport is molecular transport. It says in active transport, small molecules and ions are carried across membranes by proteins in the membrane. Energy use in these systems enables cells to concentrate substances in a particular location, even when diffusion might move them in the opposite direction. So for example, let's say we have a bunch of salt inside of our cell, and we have a little bit of salt outside of our cell. However, we need as much salt inside of our cell as we can. So molecular transport or active transport allows us to take the salt from the low concentration and move it in to the inside of the cell where the concentration is higher. And then I think our next slide is going to be a picture. So as we can see here, we have low concentration on the outside. We only have 1, 2, 3 molecules opposed to 1, 2, 3, 4, 5. So usually these 5 would want to come through here and go to the other side, so we would have 4 on one side and 4 on the other. However, in active transport, we're taking one from the area of low concentration and we're moving it to the area of higher concentration. So now instead of 5 and 3, we have 6 and 2. And as you can see, that requires energy. That is a big thing. It requires energy. And we have two types of active transport, I believe. Let's see, active transport. So we have two types. The first type we're going to talk about is called endocytosis. And large molecules and even solid clumps of material may undergo active transport by means of the cell membrane. Endocytosis is the process of taking material into the cell by means of enfoldings or pockets of the cell membrane. And the pocket breaks loose from the outer portion of the cell membrane and forms a vacuole within the cytoplasm. So endocytosis is basically the cell taking things in. So endo means inside, they kind of in, they both kind of sound similar there. So they have whole bunch of different methods for taking things in. We'll talk about those more specifically, but pockets break, but the main thing we need to know is that it's going to bring things inside the cell. So just know the first type of active transport is endocytosis. And we have two types. We have phagocytosis and pinocytosis. And phagocytosis occurs when extensions of cytoplasm surround a particle and package it within a food vacuole, then the cell engulfs it. Phagocytosis requires a considerable amount of energy, and this is what we are known as cell eating. So it's when the cell literally wraps around an entire thing, puts a food vacuole around it, and then brings it inside. So that is like it's eating up the entire thing. So phagocytosis is what that one is called. And in pinocytosis, tiny pockets form along the cell membrane, they fill with liquid, and they pinch off to form vacuoles within the cell. And that is cell drinking. So as you can see, they fill up with liquid from the outside. So this phagocytosis is solid materials, pinocytosis is only liquid, and it is known as cell drinking. So those are our two types of endocytosis, taking things in. We can either take things in by eating or drinking. And then our last one is called exocytosis. Exo sounds like exit, and that occurs when many cells can release large amounts of material from the cell in this process. And during exocytosis, the membrane of the vacuole surrounding the material fuses with the cell membrane, forcing the contents out of the cell. So inside of the cell, we will have a vacuole that has some things in it, some waste materials that maybe we don't want. That vacuole will fuse with the cell membrane, and in the process it just like catapults it out of there. So we might look at some more videos maybe in specific of these things. But just know that that is what exocytosis is, and that is all for this section. Let me know if you have any questions.